Good morning.
Good morning, everyone. I'll be moderating this breakout room. I think we're just going to give a minute or two for a couple more people to join, and then we'll start. Okay, great. So hi, everybody. My name is Miskana. Today I'll be moderating the breakout room for urology and gynecology. Just to give some pointers before, just a friendly reminder that each presentation should be no more than 3 minutes. And please, before each person goes to present your name, your title, your student title, as well as the title of your presentation. And as for questions, how we will be doing this is this room has, I believe, 7 presentations. So we'll let half of the room, so we'll let 4 of the presenters go, and then have a 5 to 10 minute question period for the first half of the presenters. And then we'll let the second half of presenters, so the 3 remaining, to do their talks and finish with another 5 to 10 minute question period for the second half of the presenters. Okay, great. So without further ado, I think we can go ahead and begin with Stephanie Sirmakesian, please.
Hi, sorry, just give me 1 second. Sorry, I didn't have my slides open. I thought they were going to be played for us. So I'm just opening the file. Is there any way you can put up the presentation? I don't know why my file is not opening.
Give me a second. I'll.
Yeah, sorry about that.
No worries.
If you prefer, I can start while Stephanie looks for her presentation.
Yeah, I think that will be. I think that'll be good if that's okay with you, Stephanie.
Yeah, of course.
Great, thank you, Elio.
No problem. Okay, so can you hear me properly? I'm just using AirPods. So.
Yes, we can hear you properly. Let me know when you're ready, and I'll.
Okay, perfect. And can you see my screen all right?
Yes.
Okay, so I'm ready to start.
Okay, go ahead. Thank you.
Okay, so hi everyone, my name is Alia. I'm a student in Dr. Compo's lab. So today I'll be presenting the antagonism of P75 receptor, which increases secretion of nerve growth factor by inhibiting the matrix metalloproteinase 9 activity in urothelial cells. So overactive bladder, or OAB, is defined as urinary urgency, usually with urinary frequency and nocturia, with or without urgency, urinary incontinence. And one of the main topics of our work is to understand how the nerve growth factor NGF and its precursor pro-NGF are involved in OAB. So NGF binds the TRK receptor to allow for cell survival and tissue survival, and pro-NGF binds the P75 receptor to do the opposite and induce apoptosis. And one of the main topics of our work is to understand how the nerve growth factor NGF and its precursor pro-NGF are involved in age-related OAB. So given the explanation that I said about NGF and pro-NGF, the NGF to pro-NGF ratio is crucial in determining tissue health versus degeneration. And we found that in aging females with OAB, NGF levels were decreased in their urine, whereas pro-NGF was stable, so there was a decrease in the NGF to pro-NGF ratio, and this could be due to an increase in MMP9, which degrades NGF. And in non-bladder tissue studies, we also found that alpha-2 macroglobulin exists. So we found that the P75 antagonist, known as THXB, actually increases NGF and the NGF to pro-NGF ratio in mice with diabetic voiding dysfunction. And we found that it actually increases decreases alpha-2 macroglobulin and decreases MMP9 in the same tissues. And alpha-2 macroglobulin is important because it actually inhibits the degradation of MMP9. So inhibiting alpha-2 macroglobulin and inhibiting MMP9 would increase NGF. So we wanted to know whether THXB or antagonism of the P75 receptor would increase the NGF to pro-NGF ratio through reducing MMP9 mediated NGF digestion. So we isolated primary culture of urothelial and smooth muscle cells from young mice bladders. Gene expression and microRNA levels were determined by RT-qPCR. Proteins were analyzed by immunoblotting and ELISA, and MMP9 activity was measured using an enzymatic kit. And we found that smooth muscle cells have a higher level of MMP9 within the cell compared to urothelial cells. And we confirmed this by measuring the inhibitory microRNA, which inhibits MMP9. So you can see that smooth muscle cells had lower levels of this inhibitory microRNA, which led to an increase in MMP9. And the opposite was observed in the extracellular space. So smooth muscle cells had low MMP9 compared to urothelial cells. And we also confirmed this by immunohistochemistry. Now, when we treat cells with P75 antagonist THXB, we actually didn't see an effect within the cell on NGF to pro-NGF. We did, however, see a decrease in MMP9 and alpha-2. And extracellularly, when we treat the cells, urothelial cells, we do see an increase in NGF to pro-NGF, which is good because it's the opposite of disease. And this could be due to decrease in MMP9 and an increase in alpha-2. And we didn't see any significant results with smooth muscle cells. So in conclusion, urothelial cells appear to be the most major source of secreted MMP9, as we saw here, and that THX potentially targets urothelial cells to increase the NGF to pro-NGF ratio extracellularly, and therefore THX could be a therapeutic tool to improve OAB. So thank you to my lab, and I look forward to questions.
Thank you, Alia. As I said earlier, I think we'll be reserving the questions after three more presentations. But thank you again for that wonderful talk. Moving forward, Stephanie, I wanted to ask if you're ready.
Yes, yes.
Great.
Can you see my screen?
Yes, we can see it clearly.
Perfect. So hi, everyone. My name is Stephanie, and I'm a second-year master's student in the lab of Dr. Lizanne Campo. I apologize for the technical error before, but without further ado, today I will be talking to you about how the secretion of NGF is regulated by nitric oxide levels in bladder cells in vitro. So Alia previously touched on this, but our lab's previous work on the urine of patients with overactive bladder disease, or OAB, has found reduced levels of the nerve growth factor NGF, which is essential to the growth, survival, and differentiation of neurons, and stable levels of its precursor pro-NGF, which causes degenerative changes such as inflammation and cell death signaling. They also found high levels of the NGF degrading enzyme MMP9 and high levels of MMP7, which is the enzyme that cleaves pro-NGF to NGF. The enzyme's activity affects the ratio of NGF to pro-NGF and establishes a balance between cell survival versus inflammatory and apoptotic processes. And finally, they found that nitric oxide was found to increase in the urine of patients with OAB and was associated with hyperglycemia. So we wanted to determine how a high glucose environment can affect the synthesis of nitric oxide and how this influences the secretion of NGF and pro-NGF in bladder cells in culture. So we used primary cultures of rat bladder, urothelial, and smooth muscle cells. The cell extracts were analyzed by RT-qPCR, immunoblotting, ELISA, and specific enzymatic kits. The nitric oxide levels were measured by the Grice reaction. And finally, we used CRISPR-Cas9 for the knockdown of genomic MMP9. So first, we demonstrated that in both cell types, a high glucose environment increased nitric oxide levels, and this led to decreased NGF secretion while pro-NGF levels remained stable. And so overall, there was a decrease in the NGF to pro-NGF ratio, which mimicked our clinical observations. Next, we treated the cells with sodium nitroprusic acid, or SNP, which is a known generator of nitric oxide. And the treated cells showed similar behavior to the hyperglycemic conditions where NGF decreased, pro-NGF remained stable, and overall there was a decrease of the NGF to pro-NGF ratio. The literature has shown that cGMP mediates nitric oxide metabolism pathways. So in urothelial cells, SNP led to a decrease in the cGMP levels, while in smooth muscle cells, cGMP increased. ODQ, which is a cGMP inhibitor, and cGMP analogs mimic the changes observed in NGF and pro-NGF secretions, which strengthen our link between the changes of NGF and pro-NGF in the presence of nitric oxide. Next, using CRISPR-Cas9 for the genomic knockout of MMP9, we showed how the SNP treatment would affect NGF and pro-NGF levels. With the MMP9 knockout, NGF levels significantly increased in both cell types as NGF was no longer being degraded. Interestingly, when we added SNP to the knockout cells, two different things occurred. In urothelial cells, NGF levels slightly decrease, suggesting that the effect of SNP on NGF is dependent on MMP9. Alternatively, in smooth muscle cells, the level of NGF almost returns to control levels, which implies that the effect of SNP is independent of MMP9 in smooth muscle cells. Finally, in smooth muscle cells, we saw that there was a decrease in MMP7 intracellularly, which could explain the decrease in NGF levels. However, in urothelial cells, there was actually an increase in MMP7 activity despite the decrease in NGF levels. And this could potentially be attributed to a compensatory mechanism of MMP7 when high levels of NGF are degraded. So just to conclude, a high glucose environment increased nitric oxide release, which led to a decrease in NGF and the NGF to pro-NGF ratio. This secretion is controlled through cGMP signaling, increasing MMP9 activity in urothelial cells and decreasing MMP7 activity in smooth muscle cells. This all suggests that the increased glycemia observed in OAB patients could potentially be part of the pathological process of OAB. Thank you for your attention, and I look forward to answering your questions.
Thank you, Stephanie. Great talk. As I said before, we'll be reserving the questions for after two presentations. Next up, we have Aya Hutch.
Aya had withdrawn. She had withdrawn from the talk because she couldn't make it.
Okay. Okay. Well, thank you for letting me know. I think we will be moving forward with Leah Christine. Is Leah Christine here?
Yes, I'm here.
Wonderful.
1 second. Can you all see my screen and hear me properly?
Yes, to both.
All right, so hi everyone. My name is Leah Christine Demers, and I'm a master's student in Dr. David Labé's lab. I'm working on a project which aims at defining the molecular basis to diet-dependent chromatin dependencies in prostate cancer. The association between dietary fat intake and prostate cancer mortality has been extensively studied over the past few years. For example, studies involving the Physicians' Health Study and the Health Professional Follow-up Study cohorts have investigated the role of diet on disease progression. The results from both studies showed that the specific type of fat intake has a direct effect on prostate cancer progression. The impact of fat intake has also been studied in vivo using mouse models of prostate cancer. The results from these studies revealed that saturated fat intake increases tumor burden, which supports the fact that a high-fat diet influences prostate cancer progression. So it is known that saturated fat intake is linked to aggressive prostate cancer. However, the mechanistic underpinnings of this link are still elusive. So in my research project, I'm focusing on investigating the role of diet-dependent epigenetic modifications on chromatin remodeling and prostate cancer progression. Our experimental design revolves around the use of 2 custom CRISPR-Cas9 libraries, which were specifically designed to target 1,500 epigenetic-related genes. These libraries were synthesized, amplified, and sent for next-generation sequencing for quality control. We have then used these libraries to perform in vitro CRISPR screens in different prostate cancer cell lines in order to identify chromatin-related genes, which are essential for cell survival and proliferation. Our first in vitro CRISPR screen was conducted in the MYC overexpressing cell line. Our results gave us many negatively selected genes, which means that the knockout of these genes leads to a decrease in cell proliferation and survival. So for example, we had the human MYC gene, which was negatively selected. This allows us to conclude that this cell line is not only cancer initiated through MYC overexpression, but it is also dependent on its maintained expression. The AR gene was also negatively selected in our screen, which is a validated therapeutic target for prostate cancer. And we also have FOXA1, which is a known pioneer factor with an established role in prostate cancer progression. So the results from our first screen showed that this screen was technically successful, and it also allowed us to identify chromatin-related genes, which are essential in the MYC-cap cell line. In the near future, we will be performing in vivo CRISPR screens in different cell lines in order to add the saturated fat diet component to our study. In these experiments, we will have mice injected with our CRISPR libraries that will be fed either a saturated fat diet or a control diet, and this will allow us to investigate the impact of diet on chromatin-related gene essentiality. Ultimately, the goal of this project is to identify chromatin-related genes that are diet-dependent and that play a key role in prostate cancer progression. We expect that the findings of this project could be eventually translated to a cancer prevention approach, which would combine precision nutrition to personalized medicine treatments for prostate cancer patients. Thank you to all my lab members and collaborators, and thank you for listening.
Thank you very much. That was a great presentation. And now we will open the floor to questions for the previous 3 presentations. So any questions, please raise a hand and go ahead and ask. I had a question, Leah. Actually, you're talking about, I'm not very familiar with CRISPR, but when you're talking about the synthesis of the custom CRISPR libraries, you said that 1,500 epigenetic genes were going to be targeted. How are those determined? How does that work?
Yeah, so to design, so you can design your own custom library like we did, and you can also pick a library that's already been designed with genes that have already been validated or guide RNAs that have already been validated. But for a custom design like ours, you'll pick the genes that you want to target based on your hypothesis. So since my hypothesis revolves around genes that are related to chromatin mechanisms, we picked all the genes that are known to play a role in chromatin remodeling and just chromatin mechanisms that are also known to be highly expressed in cancer settings. And that's how we picked those specific genes. So they're picked based on your hypothesis and based on the literature and what you want to target in your library. You also have to have positive and negative controls in your library as well. And these can be found online. Again, a lot of genes have been validated as positive or negative controls. So that's basically how you create your custom library. Does that make sense?
Yeah, that makes sense. Thank you.
Thank you.
I have a question for Ayla. I was wondering, so you mentioned that for the MMP cellular protein content that you actually saw that it was more MMP9, I think, in the smooth muscle cells compared to the urothelial cells, and that those cells were coming from single cell dissociation. How do you make sure that you're looking at the good cell type, for this type of assay?
You mean how do we isolate smooth muscle and urothelial cells?
Yeah,
So
versus the urothelial. Yeah.
Yeah, so it's actually, that's why it's really hard for us to isolate them. As you can imagine, they're like they're interacting like very strongly in the assay, in the culture. So that's why we did several confirmatory tests where we measured the microRNAs within the cell extract. So I had mentioned that there's an inhibitory microRNA for MMP9, and it was lower in the smooth muscle cells, which confirms that what we saw, which was like a higher MMP9 expression. And then also we did immunohistochemistry to confirm that as well.
But how do you actually make sure that you're having the urothelial cells and the smooth muscle cells, right, properly separated? Do you have any markers, or is that through FACS sorting?
I did not create the cell cultures. I didn't culture it, so that would have been my lab partner. So I'm not sure exactly how he did it. So unfortunately, I can't answer that question very well.
Okay, so you performed the qPCR on cell extract that was provided to you.
I didn't make the culture, yes.
All right, thank you.
No problem.
Maybe
any further questions?
Maybe I can ask a question for Stephanie. So what is the actual clinical significance of your findings? Could you elaborate a little bit on that part?
Sorry. Yeah, so like I said, the basis of this project was we had a PhD student previously looking at this, not specifically this, but looking at OAB patients from a clinical standpoint. And he is the one actually who found that there was a correlation between the increased levels of nitric oxide in the urine samples of the OAB patients, and the patients were hyperglycemic as well. So he had done some statistical analyses and had found that there was a direct correlation. And so we wanted to look at where nitric oxide fit in this pathway. And I mean, Alia had mentioned earlier, understanding this balance of NGF to pro-NGF in overactive bladder disease is crucial to understanding bladder health when it comes to overactive bladder disease. And so, it's really just to push it further and see what components are working on the MMP9, the MMP7, the NGF and pro-NGF levels. Nitric oxide, we know, is actually an activator of MMP9. So without getting too technical, but it directly leads to the activation of MMP9, which, as we both mentioned, the increased MMP9 leads to that degradation of NGF, and the low NGF compared to the stable levels of pro-NGF is where we see disease.
All right, thank you very much.
Thank you.
Thank you very much for the questions. I think we will move forward with the second half of the presentations. Thank you again to the three presenters. All right, moving forward, we will be having Florentin Rakovitan presenting the management of elderly patients with endometrial cancer.
Just a moment, please.
No worries, when you're ready.
Can you see my screen?
Yes.
So I'm Florentin. I'm second-year master student, and I'm here to talk about the management of elderly patients with endometrial cancer.
Sorry, Florentin, to interrupt you. You might want to put it in presenter mode.
I'm
At the bottom right, where you see the 64% to the left of the minus sign,
You have a little.
that's presenter mode.
Yeah, a little
A
screen.
little
There.
icon. Or you can just go to slideshow.
Is better now?
We can still see.
Please
still
just go to the lower right of the screen, just next to the Zoom place where there's the 64%, as it was mentioned.
I don't have it. The bar is on the upper corner. I don't have anything on the lower corner.
No, the bottom corner above your time, above the 1218, there's a 64%. And there's
the bottom.
There's nothing on the bottom. Everything is on the top.
But the bottom of the PowerPoint.
the bottom of the PowerPoint.
Oh,
Yes,
okay.
exactly. And then to the left, no. Left of it,
Yeah.
there's an icon to the left of where you just press the negative sign. There.
Oh, jeez. So,
But then you have to switch, right? Because we're actually seeing the is it in the display setting?
Yes, if I may, you just have to go back into slideshow where you were.
okay.
Then you click on display settings at the top, the very top, you see display settings. Next to it, yeah, there you go, here. And then you'll see if you click on display settings. Can you please click on display settings here?
I don't see display settings.
Okay, it's right where your mouse is right now.
It's on mute, my mouse. Oh,
Okay. There you go, here. And you, yeah, exactly. You click that swap presenter, view and slideshow. There you go. That's good. You're good to go.
okay, sorry about that. So my name is Florentin. I'm a second-year master student, and I'm here to talk about the management of elderly patients with endometrial cancer. Endometrial cancer is the most common malignancy among gynecology cancers in North America and developed countries. With a global increase in number of elderly patients, the number of patients diagnosed and surviving endometrial cancer has increased as well. Because elderly patients with cancer are underrepresented in clinical trials, therapeutic guidelines are merely based on data from younger patients. Standard treatments are not often well adapted to elderly patients due to multiple comorbidities and frailty. The poor survival data in elderly patients compared with the younger ones may in part reflect these treatment decisions. The objectives of this study, the primary objective, sorry, I don't know what this is happening now.
Can you explain what's happening? Can I help?
Yeah, I think it's the recording and it's the sound that's.
We can hear you. We can hear you clearly.
Okay. So the primary objective of the study is to compare overall survival, disease-free survival, and cancer-specific survival in elderly women age 70 and over with endometrial cancer and compare it with the younger patients. The secondary objective is to evaluate the treatment patterns based in women age 70 and older and compare it with younger women with endometrial cancer. The third objective is to evaluate the trend of survival data in elderly patients with endometrial cancer and explore factors related with these changes. And the fourth objective is to retrospectively classify patients with endometrial cancer using frailty markers and to use regression analysis to evaluate the predictive value of these markers on overall survival. A few words about the methods. Study design is a single institution prospective study conducted at the Jewish General Hospital in between 2003 and December 2019. Study population, all consecutive patients over the year of 18 years and older with endometrial cancer treated in this institution. Sample size approximately 1,500 patients. Regression analysis will be used to control for confounding factors. Survival curves will be generated using Kaplan-Meier graphs and compared with the log-rank tests. And for time to events, Cox proportion hazard model will be performed to calculate hazard ratio with the 95% confidence intervals. Interim results. 1,060 patients treated at the Jewish General Hospital between 2009 and 2019 have contributed to this data analysis. 730 patients below the age of 70 and 330 patients age 70 and older. Overall survival at 5 years was 92% for the younger patients below the age of 70 and 80.1% for the patients age 70 and older. The difference in survival between the two groups was statistically significant. However, no statistically significant difference was noted in 5-year disease-free survival between the two groups. And you can see the graphic on the right here, the survival curves, they're almost identical for recurrence rates and disease-free survival. In conclusion, similar recurrence rates between the two groups showed that age alone was not a significant predictor for disease-free survival in our study population. Detailed evaluation of the association with tumor characteristics, patient frailty, and therapeutic approaches is currently underway. Thank you, and sorry about the technical issues.
Thank you very much, Florentine. Thank you for your patience. We'll reserve the questions for afterwards and move forward to our next presenter, Stephen Serafini. Oh, Florentine, if you could stop sharing your screen, please. Thank you.
Hello, can you hear me?
Yes, we can hear you. Excellent. And we can see your presentation.
Excellent. All right. So hello, everyone. Today I'll be presenting my latest progress on my project titled Participation of Sphingosine and Theramine in Human Burn Capacitation. Oops, it's not changing slides. There we go. So here's a brief outline for today's presentation where I'll provide you a background followed by hypothesis objectives, methods, results, and lastly, my conclusions. So onto the background. So first and foremost, the clinical relevance of my work is that we opt to further characterize the mechanisms that lead to idiopathic male infertility. Now, idiopathic male fertility is defined as the unexplained reduction of semen quality or men with normal semen parameters but are infertile. Now, idiopathic male fertility represents 34% of all male infertility cases as depicted on the figure on the right-hand side of your screen. Now, moreover, sperm capacitation is not evaluated in idiopathic male infertility. Is not assessed at the fertility clinics to evaluate infertile men. And so what is capacitation and what is the role of single lipids? So firstly, capacitation is a tightly regulated process that includes biochemical and morphological changes of spermatozoa and is essential for the acquisition of fertilized capabilities. Now, single lipids represent 20% of the total membrane lipids in spermatozoa. Sphingosine, ceramide, sphingosine 1-phosphate, and ceramide 1-phosphate are bioactive molecules that have been demonstrated to be involved in signaling. However, the role of lipid signaling in sperm capacitation is largely unknown. And so we hypothesize that sphingosine, ceramide, sphingosine 1-phosphate, and ceramide 1-phosphate are necessary to regulate the production of reactive oxygen species and phosphorylation events during human sperm capacitation. Our objectives are to first determine the requirement of sphingosine and ceramide or their phosphorylated forms in capacitation. Second, to determine the presence of the sphingosine 1-phosphate receptor 1 in human spermatozoa. And third, to determine whether the sphingosine 1-phosphate receptor 1 signaling activates phosphoproteins associated with sperm capacitation. So briefly, materials and methods. So upon attainment of healthy human semen samples, they will be undergo liquefaction and isolation of highly motile sperm by centrifugation prior to manipulation. Then they'll be incubated with or without fetal cord serum ultrafiltrate, FCSU, which is a known inducer of capacitation, in the presence or absence of various inducers or inhibitors at 37 degrees Celsius for 4 hours. Immunoblotting will enable the detection of varying levels of protein in our samples, while immunocytochemistry will then enable the detection of the receptors and related activity. So now onto my major results. So first, presented in front of you is an immunoblot of antiphosphatyrosine and its corresponding relative intensities down below. And what can be seen is that first, we have an increase in antiphosphatyrosine signal with fetal cord serum ultrafiltrate versus without FCSU, indicating that the sample capacitated. The major finding of this slide, however, is that sphingosine at 40 micromolars and ceramide at 40 micromolars. I feel like I can't see. Oh, there we go. There's my cursor. Sphingosine at 40 micromolars and ceramide at 40 micromolars increase the amount of phosphatyrosine signal relative to the negative control, thus indicating that both induce capacitation. Second, what was found presented is another immunoblot of antiphosphatyrosine and its corresponding relative intensities, and it was demonstrated that by inhibiting sphingosine kinase 1 with sphingosine kinase inhibitor, which sphingosine kinase is responsible for converting sphingosine to sphingosine 1-phosphate, as well as inhibiting ceramide kinase with ceramide kinase inhibitor, which converts ceramide to ceramide 1-phosphate with increasing concentration. And when added together, we then have a corresponding decrease in the amount of phosphatyrosine signal, which is then further depicted with the relative intensities down below. Now, this demonstrates that sphingosine 1-phosphate and ceramide 1-phosphate are essential and required for capacitation. Moreover, what we have in front of you are the immunocytochemistry results and a figure demonstrating the percentage of spermatozoa with 1 or 2 of sphingosine 1-phosphate receptor 1 patterns. So what we found is that sphingosine 1-phosphate receptor 1 in non-capacitated spermatozoa, this receptor is found predominantly throughout the entire head, whereas in capacitated samples, it was found towards the post-acrosomal region of the sperm head, as well as the equatorial segment. Moreover, and lastly, we have found that sphingosine at 40 micromolars was found to increase this is an immunoblot of phospho-PI3 kinase, and what was found is that sphingosine at 40 micromolars was found to increase the amount of phospho-PI3 kinase signal relative to the negative control without FCSU, excuse me, and that VCP23019, which is an inhibitor of the sphingosine 1-phosphate receptor 1, was found to decrease the phospho-PI3 kinase signal as shown in the immunoblot. Now, this shows that sphingosine and its receptor are involved in downstream signaling and engage with phospho-PI3 kinase AKT pathway, leading to subsequent nitric oxide production, which is important for sperm capacitation. Now, in conclusion, sphingosine, ceramide, and sphingosine 1-phosphate and ceramide 1-phosphate have been shown to be involved in capacitation. And sphingosine promotes phosphorylation of phospho-PI3 kinase, which is important for nitric oxide production during human sperm capacitation. Now, the significance of our work is that we opt to increase our knowledge of lipid signaling during human sperm capacitation, which will then help us to understand this complex process and allow for the development of novel diagnostic tools and treatment for male infertility. Thank you.
Thank you, Stephen. Moving forward with our final presentation, we have Michelle Shen.
Okay. Okay. Can everyone see my screen?
Yes, we can see and we can hear you clearly.
Okay, perfect. So good morning or good afternoon. My name is Michelle. I'm a second-year MSc student working with Dr. Labbe. Today I'll be discussing my project looking at the role of FOXA1 in MYC-driven and obesity-dependent prostate cancer outcomes. So obesity is a fairly prevalent health concern in Canada. It's estimated to affect about 63% of the adult population. Prostate cancer, likewise, is the most commonly diagnosed cancer among Canadian men, not including non-melanoma skin cancers, and it is the third leading cause of cancer-related death. Now, at the intersection of these two, as my lab member Leah outlined before, there's some pretty good evidence that diet-induced obesity is associated with aggressive prostate cancer outcomes. And what we don't really understand are the molecular mechanisms that underlie this association. So we do hypothesize that chromatin regulation and epigenetic reprogramming may drive diet-induced obesity-dependent aggressive prostate cancer. And in my project specifically, I'm interested in the role that FOXA1 and the role that FOXA1 might play in this dynamic. So to look at the role of FOXA1, we did knock it out using CRISPR-Cas9 in a MYC-driven murine prostate cancer cell line, and we did validate this knockout using a Western blot. And we looked at the effect of FOXA1 knockout in vitro as well as in vivo using the allograft model. Now in vitro, we did see that FOXA1 knockout attenuated cellular proliferation. So the growth curve on the left shows that FOXA1 knockout cells do indeed grow slower relative to unedited controls. Similarly, the box plots on the right show the maximum growth rates that these cells can achieve in a variety of serum concentrations. Again, we see that FOXA1 knockout does seem to impair or attenuate the maximum growth rate that these cells do achieve. Now in vivo, we want to see whether FOXA1 contributed to MYC-driven tumor growth, and specifically we were interested in whether there are any diet-dependent outcomes on tumor growth. So we kept mice on either a high-fat or controlled diet, and we injected these mice with tumors or cancer cells where FOXA1 was either knocked out or where FOXA1 remained unedited. Now, interestingly, we did see a diet-dependent effect on the time it took for these tumors to reach a measurable size. So specifically in controlled diet-fed mice where FOXA1 was knocked out, we saw that there was a significant delay in the time it took for their tumors to reach a measurable size. We did not see this effect in the diet-induced obesity mice. Now, when it comes to the average tumor growth rate, there were a couple of things to note. First of all, when comparing the control diet to the diet-induced obesity arms, we do see that diet-induced obesity seems to drive more aggressive tumor growth, and that is in line with epidemiological evidence. More interestingly, when we compare the FOXA1 knockout to the unedited arms, we see that FOXA1 knockout itself seems to impair tumor growth. And interestingly, if we compare the diet-induced obesity FOXA1 knockout arm, we see that FOXA1 knockout seems to attenuate the growth advantage that diet-induced obesity seems to confer in this condition. Now, to more conclusively, I guess, determine the interplay between diet, FOXA1 and cancer progression, we did also conduct RNA-seq on our FOXA1 knockout cells in vitro, and interestingly, glycolysis was a negatively enriched hallmark in our FOXA1 knockout cells. And similarly, in vitro, we also did see that FOXA1 knockout seems to impair basal mitochondrial respiration. So this suggests that FOXA1 does contribute to metabolic regulation. However, how this manifests in vivo in response to diet is still to be determined. So in conclusion, we've shown that FOXA1 contributes to MYC-driven prostate cellular proliferation as well as tumor growth. FOXA1 knockout seems to delay tumor formation in controlled diet-fed mice only. And then again, molecular profiling of these tumors will reveal whether there are any specific metabolic pathways that interplay with FOXA1 in order to drive obesity-dependent prostate cancer progression. And we do hope that the identification of these diet-dependent responses will illuminate how we can consider dietary intake and leverage it when treating prostate cancer patients. So thank you very much for listening to my presentation.
Thank you very much, Michelle. Before moving to open the floor for questions, I just want to remind everyone that the link to the evaluation forms for this room, for the session, are in the chat box. And now I open the floor to any and all questions.
Maybe I can start with a question to Stephen.
Yes.
So you mentioned that the ceramides were prevalent in the membrane lipids of the spermatozoa. So what is the specific ceramides that are actually part of that lipid membrane?
Well, from my understanding, there's various forms. From what we never actually analyzed the specifics for which ones are present. However, for our experiments, we are using C6 ceramide and for inducing capacitation. If that is if that answers your question.
Okay, so you're using a ceramide that you don't know whether or not it's important in that specific model.
From my understanding, because not much has been done on spermatozoa or human sperm specifically as well, so it's quite, I guess it's quite novel, and so in the future, I believe like it would have to be further depicted. However, in other cell types like somatic cell types, C6 ceramide has been it has been demonstrated to be present in other forms as well. And so, yes, I believe there's much work to be done in terms of further elaborating on which ones are present and which ones are active and whatnot.
All right, thank you. Maybe I can ask a question for Florentin as well. So you mentioned that disease-free survival was not driven by age, right? So what do you think is the clinical pathological data parameters that is actually important for disease-free survival in your patients?
Yeah, so very good question. There are several factors. Based on the literature, my understanding is that elderly patients with endometrial cancer have more aggressive forms of cancer, higher grade of cancer, which leads to a poorer survival. But also, again, based on data, what I found in literature is that they're often undertreated. Patients, physicians, surgeons are concerned about these older patients with comorbidities and frailty not being able to tolerate the standard treatment, and they end up being undertreated, which again leads to a poorer survival. It may be other factors. This is the reason why we're looking into almost 20 years of data, but these are the two things that come to mind.
Right, thank you.
Any further questions for our three presenters or even the three from the first half?
If not, maybe I can ask a question to Michelle, but I know where it's going. So what would be the next step to your project?
So as I mentioned, there's a fair amount of molecular profiling to be done, and along with that, we would like to epigenetically profile these tumors to see more precisely like how diet affects the epigenetic profile. As we mentioned, like that is part of the original hypothesis driving this project. And then of course, another interest in research avenue would be to look at the role of FOXY1 overexpression in this model as well. We do know that FOXY1, I guess, aberrant FOXY1 activity and overactivity is more relevant clinically. That's what we see. So how that also intersects with diet would be an interesting avenue to explore.
All right, thanks.
Thank you for the questions and the presentations. I'll once again remind everyone that the link to the evaluation forms for this room are in the chat box. So please follow that link. And if there aren't any other questions, I think we finished several minutes early. But I believe from 12:50 to 1:15, there is a break for lunch, and we will reconvene for the fourth session steered by Dr. Derek Rosenzweig. And yes, thank you again to all the presenters. They were wonderful presentations.
Hi to all of you. So you could click on leave room, the blue button, and join us into the main session. Thank you.